So here we're going to understand the oxidation of propionyl-CoA, but what is propionyl-CoA? Where is it coming from? And in order to understand that, we need to take a step back to beta oxidation. Remember that when we talk about the breakdown of fatty acids for energy, we utilize this process known as beta oxidation. And the product of beta oxidation is our acetyl-CoA. So what essentially happens is that we have our fatty acid and it's going to be a varying length of carbons, and we break it down into two carbon molecules, and those two carbon molecules are known as acetyl-CoA. Now, we can imagine if we have an even numbered carbon, let's say we have 1, 2, 3, 4, we have 4 carbons. The first two will cleave off into acetyl-CoA, the second two will cleave off into acetyl-CoA. So this can be broken down perfectly. But what if I added another carbon over here? Now, the first 2 carbons over here become acetyl-CoA, and if I try to break these next 2 down to get acetyl-CoA, I'm going to be left with 1 carbon. So essentially, when we talk about odd-numbered fatty acids, the final round of beta oxidation, instead of giving us acetyl-CoA, which is a 2-carbon molecule, we get a 3-carbon molecule, and this 3-carbon molecule is known as propionyl-CoA. And to break down propionyl-CoA, we have a different pathway, because remember, acetyl-CoA, once we gain that from beta oxidation, acetyl-CoA is going to be fed into the TCA cycle, where it's going to produce FADH2, it's going to produce NADH, which are electron carriers, which are going to go into the electron transport chain to produce ATP through oxidative phosphorylation. But we're not looking at acetyl-CoA. We have to break down this 3-carbon molecule known as propionyl-CoA. And how do we do that? It's a simple step reaction. So first we have to convert propionyl-CoA into dimethylmalonyl-CoA. How do we do that? Well, we utilize biotin, and biotin is actually a really important cofactor, and we'll look at the mechanism of biotin in a separate video, how it plays its role. But this reaction is catalyzed by propionyl-CoA carboxylase, and essentially we have this bicarbonate molecule, and we're utilizing ATP, and we're going to produce ADP and inorganic phosphate as a result. But essentially, we take this HCO3, and what we are doing is this bicarbonate, through the mechanism of biotin, is what's responsible for attaching this carbon dioxide group onto our propionyl-CoA at that second carbon to produce dimethylmalonyl-CoA. But what do we want to do next? We actually want to isomerize this dimethylmalonyl-CoA. How do we isomerize? Well, we need an isomerase enzyme, and we are using an epimerase. So we have methylmalonyl-CoA epimerase, which is going to move this coenzyme A group over here to this carbon over here that we've just added. So we can see that switch. The coenzyme A group, it moved from that 3rd carbon onto this carbon that we just attached. And as a result, we get L-methylmalonyl-CoA. Now this was an isomerization reaction. It was an inversion of our stereochemistry. Now in our final step, what are we going to do? We're going to utilize a mutase, and a mutase is responsible for moving a functional group. And specifically, it's methylmalonyl-CoA mutase, which is our enzyme. And once again, we have another cofactor. It's coenzyme B12, which also has a special mechanism, which we will look at in another video. So we've gone over 2 cofactors. We have coenzyme B12 over here and biotin over here. And if we understand the mechanism behind both of these, we can understand how the change in isomerization and the functional group movement is happening. So back to our L-methylmalonyl-CoA, what are we going to do? We're going to simply move a functional group, and we can see that this carbon group over here with the coenzyme A is being moved from that second carbon to this carbon up top over here, and as a result, that second carbon now bears a hydrogen. Now, what is this molecule? This molecule is Succinyl-CoA. Where have we seen Succinyl-CoA before? Succinyl-CoA is an intermediate in the TCA cycle. So this Succinyl-CoA, it's going to enter the TCA cycle, and it's going to move through the TCA cycle, and it's going to move through the anaplerotic reactions as well, because remember, we want a balance of the intermediates in our TCA cycle. So it's going to move through an anaplerotic reaction. It's going to be fed back into the TCA cycle as acetyl-CoA, and it's going to be utilized to produce electron carriers, which are going to produce energy through the electron transport chain. But essentially, this is propionyl-CoA oxidation. It's that 3-carbon molecule at the end of beta oxidation in odd-numbered fatty acids, which is broken down into succinyl-CoA, which is going to go into the TCA cycle to produce energy.